Good morning, everyone. I'm Etienne Bilgo from l'Institut de recherche en sciences de la santé, IRSS San Muraz in Bobo-Dioulasso, Burkina Faso. It's really a great honor and pleasure to me to present you this morning some data from my thesis work. The title of my presentation is The Transgenic Insect Killing Fungus Better Kills Insecticide-Resistant Malaria Vector Mosquitoes. As you know, after 40 years, the most important weapon against mosquitoes may be failing, remain insecticide. Because mosquitoes are developing resistances in most of the countries where malaria is endemic. And we have three main types of resistance: the biochemical resistance, the behavioral resistance, and the physiological resistance. So that makes some entomologists caricature this situation by saying that mosquitoes getting stronger and smarter. So we need urgently new tools and complementary tools to support the common tools actually used to fight malaria. In the nature, fungi cause the majority of disease of insects, particularly fungi from the genus Beveria and Metarhizium. You can see in the screen some orders of fungus that from the infection of some orders of insects that from the infection from fungi. And here, this is the mosquitoes dead from the infection of Metarhizium fungus. How fungi infect insects? Fungi infect insects actively by direct contact with the cuticle, and their works as the common insecticide that we use. When the spores fungus enter in the contact with the cuticle of the insect, they go through the different layers of the cuticle, and after 30 to 72 hours, when the spore reach the blood, the hemolymph of the insect, they grow a lot, and a couple of days later, they cause the death of the insect. When we try to use this fungus to tackle malaria, specifically the fungus Metarhizium, which is one of the most virulent fungus against mosquitoes, that takes about 10 to 2 weeks to kill mosquitoes. So that can be a problem if you want to use this fungi to fight malaria, because fungi have enough time to go to mate, produce eggs, contribute to the next generation, but also the mosquitoes have enough time to blood feed and transmit malaria. So Professor Raymond Saint-Léger from University of Maryland at College Park proposed since 2007 to transform genetically the fungus in order to increase the efficacy of the fungus. He introduced in the genome of the fungus a neurotoxin called hypertoxin. This toxin came from a blue mountain spider. And these toxins are well known, and we can synthesize the sequence of genome that code for the production of this toxin. And this sequence of code are introduced on the genome of the fungus. And we already ran lab assays against lab-bred mosquitoes, and the result were so promising. But we don't have any data against wild mosquitoes in the condition closer to the reality. So the ultimate goal of our studies was to evaluate the efficacy of this transgenic fungus against wild-caught mosquitoes in the semi-field condition. So first, we want to find the best method to infect mosquitoes in the field, but also make bioassays in the lab and in the semi-file against wild-caught mosquitoes. And the parameters we evaluated was the mortality, the survival, the impact of the infection on the blood feeding, the fecundity, and the fertility. The design of our study consisted firstly by larvae collection in the field here at Vallée du Cou in Burkina Faso, and then we brought the larvae in the insectary that we breed them, we identify them firstly, breed them, and we use adult for bioassays and observations. So in the same time, we proceed to the culture of the fungus. So the strain that we used for our bioassays were Metarhizium hybrid strain. This is a transgenic strain coded with the green fluorescent protein, and the natural strain coded with the red fluorescent protein. And we also isolate local strain of Metarhizium from Burkina Faso that we use for bioassays. And we ran bioassays also with common insecticide used in Burkina Faso. And we ran bioassays in the lab and in the greenhouse in Burkina Faso that we built during this project. As a result, when you compare the efficacy of these fungus with a common insecticide, we can see here that the bioassay in WHO tubes, 1 hour contact, and we make observation. Instead to make observation during 24 hours, we make observation for 7 days. And we have this result. We have around 100% of mortality with Kishimu, which is the WHO sensitive insecticide strain, and we have 100%. This is not quite surprising. And we have also with hybrid strain. With hybrid strain here, we have a mortality around 100%. And for the natural strain, we have around less than 75%. For this, when we run bioassays with our local mosquito strains, we can see here clearly that the common insecticide that we use for malaria control here has a mortality around 10%, not different from the control. But the transgenic strain has a mortality around 100%. And the conventional strain, the natural strain, has around less than 75% of mortality. When we evaluate the impact of the infection of the fungi on the blood feeding interest in the tunnel test, you can see here clearly that with the transgenic strain in green line, we have by day 3, less than 60% of mosquitoes are interested by the blood feeding. And by day 5, we have around 40% of mosquitoes still interested by the blood feeding. But if you compare that line with the natural fungus, we can see clearly that by day 3, you still have more than 80% still interested by the blood feeding, and by day 5, you have a big proportion of mosquitoes that still interested by the blood feeding. When we combine the data of mortality and the blood feeding interest, we can have in this model that you can see clearly in this model that in the green figure here, you have less than 20% of mosquitoes are interested. Less than 20% of mosquitoes are able to transmit malaria. But with the conventional strain, the natural strain, you have by day 5 more around 80% of mosquitoes that can transmit malaria. During this project also, we isolate and identify some strain of Metarhizium and other entomopathogenic fungus, and we run bioassays against mosquitoes and the non-target insects like grasshoppers and bees. And you can see here that we have above this isolate, we have two promising isolates that we call for the moment S10 and S26. They have a virulence, their virulence are higher than our natural fungi that we use for bioassays. But we're still working on that to understand the mechanism and the toxin that they produce and the molecular identifications. In conclusion, we can say we are here in the phase 1 and phase 2 of this project. We're trying to make bioassays in the lab, in the semi-field. But moving from this step, phase 1 and 2, to the next step, this is the field, open field studies. We have to fill certain conditions, obligatory. That means the condition of efficacy and safety of the product, and obtain all of the approvals and permits from the authorities, and also work closely with the local population, like the stakeholders, the local villages, and the administrations. I want to end my presentation by thanking some institution persons at the NIH for funding these studies, and University of Maryland, College Park, for Professor Raymond Saint-Leger, Brian Lovett, and Dr. Shaoling Li. I want to thank also Professor Marcelo Jacobs Lorena from University of John Hopkins, my advisor at University of Ouagadougou, Professor Antoine Sano, and all of my co-lab workers from IRSS and Saint Muraz. I want to finish to thank my closer advisors, Professor Roch Dabire, and my mentor, Dr. Abdoulaye Diabaté. Thank you for your attention.